later. Okay, so how we're gonna do for today from today's lecture is I'm gonna go through just a lecture slides because there are small little questions. I'll ask you guys to go through one by one, quickly go through, and then we come back to the YouTube where you pose these questions, and from there we'll start our discussions. So hopefully this is a new trial, but I think this is would be the most beneficial because I know you guys will spend time for watching video. So if possible, we will try to finish the class time shorter to save your time. So that's our plan. So let's get started. Let's see. First, I will share the screen with you, and then let's go through one section by one section. Okay, let's see. Share screen of medical engineering. Let's see. Okay, I'll just share my whole screen with you. Okay, that might be a good way. Okay, can you guys see the screen well? Raise your hands. Okay, good. Let's see. I want to see all of your face. So instead of playing the movie, which you are supposed to already watch it, let's go through from the beginning. So this is a biomolecular principle which you are supposed to have. Majority of you, I think, have the background, but for those who may be just pure engineering background may lack of some of this. So I'm gonna go through very quick with you. So let's see. So this is homework related second homework. So the homework is usually not a lot of amounts because I think it's 200 to 250. It's a pure essay. Don't try to summarize all the article content, which is you're supposed to read through, but more important thing is what you have gained, a new insight, what you have felt. And that's the most important. Don't consider there's like one one standard answer for this. This is an essay, but I do want you to go through and then think about what has happened and what kind of lesson you got, like personally. All right, so let's go through. So today, we'll go through molecular principles in BME. So the learning objectives, there are many, but we want to start from the most basics. So basic structure of proteins, and which is polymers of amino acids, and how diversity of amino acid structures influences protein three-dimensional structure and function. So like we all call like DNA. We all know that that's very important. I think I wrote some stuff down. We can go through later. And then building blocks of a protein, which is so-called amino acids and polypeptides. So I'm going to just go through with you one by one. So let's see, Anho, proteins are made up of
polypeptide.
Okay, great. Yes, polypeptide chains. So I think I can, let me see, like everyone probably knows. And second, Feng, what is a polypeptide? Polypeptide is polymers of, we can't hear you. You have to turn on.
Amino acid.
Exactly. Nice. So we already know the amino acid structure, which is a carbon base and hydrogen and amino and carboxylic groups. And this R group, which determines that 20 different kinds of various types of amino acid. So, okay, so I think that's good to go through. And this slide is showing you a category of the property of the amino acids, which is nonpolar, polar, and acidic or basic, and that's total 20. So many proteins are, let me see, 정우주.
Yes, Professor. Yes, Professor.
Yes, many proteins are N something catalyzing chemical reactions. Can you guess?
Enormous.
Find that, Youngmin?
Yes, enzyme.
Excellent. Yes, many proteins are enzymes which catalyze chemical reactions. So this is a kind of a protein which is kind of micro machine doing something, right? But other proteins may be a little different, which is a ST support inside and outside of the cell. Jung-woo, again, can you try to consider?
Structural.
Exactly, great. Yes, other proteins are providing structural support, such as ECM or extracellular matrix. So proteins does, many proteins and enzymes, which is micro machines, catalyzing chemical reactions. The other kind of proteins are simply structural proteins, such as like collagen. Okay, so I think you're good on this. Let me see. And then this slide is actually showing you to get into more details for especially into biochemistry. Okay, so that's I just put it there, and this diagram shows how to make a polypeptide chain reaction, which kind of results in a dipeptide plus a water molecule. All right, so the next is probably one of the most important. Like we do this, ask a lot about, especially like entrance exams and those. So protein structure and function is starting from a single amino acid as a basic element, making a polypeptide chain, and somehow in the end we've got this nice three-dimensional functional unit of structure. So we divide this one into first, second, third, and fourth in more jargon way, like primary, secondary, tertiary, and quaternary structures. So primary structure is, you guys can see, let me see, Doreen, can you guess what is this primary? Is a chain of amino acids. Doreen, we can't hear you.
I don't remember, sorry.
Oh, that's fine. You know, chain of amino acid, I say, chain of amino acid, there are 20 different kind of amino acids. So look at that, asparagine, glycine, phenylalanine, I don't know, like this, glutamic acid, alanine, arginine, aspartate. So these are like kind of going, where are the information? And that information is this sequence of a chain. So that we call as a primary structure. Doreen, is it clear?
Okay. Yeah.
Okay. Yeah. So once we make like a thread, like a chain of amino acid, now you have to form a kind of a structure from here. And the important thing of this amino acid is you see the residual, which determines the polarity or chemical like pH or various chemical, biological functions and characteristics. So with that, we actually realized there's two kind of functional units, which we call secondary structure of protein. So that has two different kind of distinctive shapes are coming up. So the first is called the alpha, Shin-So-yeon?
Oh, yes, Professor. Alpha helix.
Exactly, yes, helix. So somehow the chemical structure of this is kind of, maybe repeating and making a nice helical structure that we call alpha helix structure. And the second is like, looks like this, that beta Pleiades, let's see, Lee Jung-yoon.
Beta plate sheets.
Great. Yes, sheet. It's like it's your some of your clothing thread. So it looks like this. Okay, so that's better. Please, I describe it here as you look at this. These ones looks like helical structure. The other ones like this way going this way, and the other this way, the other this way, the other. And their corresponding one has a nice, interesting interaction, which make a structure of this. So that we call a beta pleated sheet. A pleated is like in terms of figure like this. So once we have these two basic units, now you can actually combine this together into a nice three-dimensional structure that we call tertiary. So as I say, this is 3D arrangement of secondary structure with, let's see, Janice?
One polypeptide.
Exactly, very good. So like you see the protein, tertiary structure is a one polypeptide chain here. So one, you make this kind of magical structure, that's a tertiary structure. But sometimes protein can be just one polypeptide chain, but other times some proteins are like performing a complicated function which will need a multiple subunits. In that case, we call it as a quaternary structure. So all polypeptide within a complete protein with a multiple, of course, subunits. A good example is hemoglobin molecule. You can see these 4 different kinds of colors, which is they have 4 subunits to perform hemoglobin function, which is a grab an oxygen molecule and go down to the all the tissue part and then release the oxygen to where it's necessary. All right, so we look at a little inside protein structure and function. And as I say, if you go into inside primary structure, eventually it's a single amino acid has a carboxylic end, and the other is a amino end. So we call this as a C terminus and N terminus. And you stack this in the end, one side is a C terminus and the other is N terminus. So always the sequence of a primary structure has C and N terminus in either side. And the secondary structure you can see with this dot here, this is actually a hydrogen bonding, a partial positive and partial negative. They attract each other to form and exert a force to make a nice helical structure. And the other interesting is, as you see, this arrow means because primary structure has from N terminus and C terminus. So that's why here this way, and the other, you can see this hydrogen bonding formation. This makes a nice pleated sheet structure. So these two are called secondary structure. And once you make this combination, you'll have a tertiary structure which can hold, in this case, hold one heme molecule, but at the quaternary structure, a full hemoglobin molecule have these 4 subunits. They are connected together, and you can see they can hold 4 heme molecules. Okay, so that's like what I said, home exercise. So this is the first one, and I want to let me stop share, and then bring here. Let's see if you have any questions here. Okay, there's no question. So I think we'll just move on, right? Let's see. The second one. Oh, okay, there's some questions here. So let's see. Are you guys seeing the screen or not? Let me share the screen. Okay. So let me share the screen. Can you see it clear? Let's see. Let's make it big so that you guys can see it more clear. Okay. Raise your hands if you can see the screen clear enough. Good. So interestingly, there is some questions. So Mahanur Zahir. Okay, where's Mahanur? Mahanur, can you show your face? Sorry, I can't see you, but probably you have some reason. So let's go through. What is the role of, okay, good. What is the role of hydrophilic and hydrophobic interactions in the structural configuration of protein, specifically alpha helix and beta repeated sheets? Oh, very, very good question. Hydrophilic and hydrophilic interaction. It's a good question. Let's see. Is there anyone who can try to answer this? Biochemist here. Let's see. 정우주, any guess for you? Is this a question clear or? You can turn on your mic.
Actually, I couldn't understand clearly this question, but Mahun just interesting about how is different the structure of between hydrophobic and hydrophilic interaction.
Yeah.
So I can't explain how to different about alpha helix and beta splitted sheets with hydrophilic and hydrophobic. I just can say hydrophobic is unlike the water environment, and
Yeah.
hydrophilic like water environment.
Okay, so yeah, it's a little unclear. It could be a little. So hydrophobic, I think it's a typo, probably hydrophobic and hydrophilic interaction. So, you know, this is good to have a question you guys can answer, you know, even later on, okay? So let's go through the second question of Youngmin. Insulin is produced from one polypeptide, and the intermediate peptide is cleaved and looks like two polypeptide. Is it a tertiary or quaternary structure? Oh my gosh, this is very, very good question. Okay, Youngmin, so there are some people who put some answers, which is great. Let's see, Anho, I read about difference between among 4 protein structure and primary, secondary, tertiary. Okay, that's that may not be a clear answer for this. I'm not sure, because insulin was produced from one mRNA and became 2 chains during post-translational modification. Okay, Youngmin, raise your hands. I love this question. Can you explain a little more of this? I mean, this is kind of an open question, but in what sense do you think this insulin molecule is 2 polypeptide putting together, tertiary or quaternary? I'm usually you can say your opinion.
I think insulin has tertiary structure because it is from one messenger RNA.
Okay. Yeah, please speak out loud. Sometimes it's not very clear to listen, but your point is very, very interesting. So insulin starts from one mRNA molecule, which will create one polypeptide chain. Okay, and then there's the intermediate cleavage, and looks like 2 polypeptides. So I love this question. You know, you guys, while doing this, you should do a Google search and then put the answer. Those will get the point. I just love this question. And honestly, I have to say, I don't know the answer, all right?
No, actually, my answer is more. You should put more.
Oh my God, okay. Sorry, I didn't know that. So made from 2 chains connected together by 2 disulfide bonds between 2 cysteine residue. So it's a tertiary structure. Okay. Yeah. All right. 2 chains connected together by 2. But the Anho. So here's a question for you. So you guys probably know, like for example, hemoglobin molecule. There's like 1, 2, 3, 4 subunits. How do they connect to each other? So the answer is disulfide bond, right? S to S, right? And so in that case, but in this case, Anho made a comment that made from 2 chains connected together. Anho, from what you wrote, I consider this sounds like a quaternary structure because two chains and then put the disulfide bond to bring them together. Then isn't it supposed to be quaternary structure?
No, because firstly, I also confuse between the terrace because before I think this is the quaternary structure, but when I read again, they say like if you want to become the quaternary structure, so you have to become the tertiary structure first, and then from there also tertiary structure, then you connect together, so it will become quaternary. But the insulin, this is not the tertiary structure. So it's just like the secondary structure connect together.
Mm-hmm.
So this will become the tertiary, not quaternary structure.
Oh, okay.
Yeah, as I understand.
Okay. Yeah, I mean, that's a good theory. I agree with you for now. So, but this is a very good question, and I would love to have you guys search and even put more of your vote on this. So for example, like this. Okay, I love this answer. And hey, I like this answer. So I'm sure there are other opinions. There's more answers. It's likely that's gonna be scientifically more accepted in our community. Okay, so I love this one. Okay, thank you. And let's move on to the next. So I hope this is still pretty good, clear. So biomolecular principles. So actually, there's a question about like this bonding by Manohur. So biomolecular principle is a basic concept, which is very important. We all know like our water molecule is partially positive in the hydrogen atom side and partially negative in oxygen side. So that this small water molecule is kind of, they actually interact each other, actually attract each other. And that we call as a, I think everyone knows this, so I wouldn't actually ask any question. This is hydrogen bonding. And now we get to this genetics aspect. It's nucleotide, which is composed of pentose, or like five carbon molecule, sugar molecule, and the phosphate and organic base. This organic base actually determines the 4 types of a nucleotide. So this is composed of actually, I already told you it's a pentose. You guys all know like pentagon in US or like this penta means a 5. So you can see 1, 2, 3, 4, 5 carbon. And this P is called a phosphate group and an organic unit, which is a organic base. Okay, so I think you guys all know this clearly. So I just want to move on. And so basic concepts of this is in fact kind of an acid. So what we call this is a, let's say, Janice, how would you call this?
Nucleic acid.
Exactly, yes, so we call it nucleic acid. I think you will find out this one in cellular nucleus, right? So that I know, I'm sure you know, like nucleotide, there's ATCG for DNA, but the AUCG for the RNA molecule. So the question is, we have studied about this protein structure coming from 20 amino acids. Now we are going into this genetic material called DNA, and DNA is made up of this nucleic acid. So that comparison would be important to distinguish the concept. So amino acid has about like diversity of this monomer in a macromolecule. There's a 20 kind of amino acid, and biological macromolecule from the amino acid would be let's see, not.
Yeah, Professor.
Can you guess what this is? What is a macromolecule made out of amino acids?
I don't know, Professor.
That's fine. Yundami, can you guess?
I don't know, Professor.
Oh, that's fine. So we just discussed about like primary, secondary, tertiary, quaternary structure made out of amino acid. And what's the final outcome of this amino acid chain, you know, alpha helix, beta, pleated sheets, and then tertiary structure and quaternary structure. And what's the outcome in the end? What's the macromolecule? Yeah, so that's a peptide. Amino acid is making a peptide. And that peptide is multiple peptides are coming together with a polypeptide. And in the end, we generate a protein molecule. Does it make sense? Okay, then what about biological macromolecule of deoxynucleic acid or ribonucleic acid? DNA or RNA is made out of this nucleotides of ATCG. How many are there? Janice, can you guess? Just count the number of here.
Five.
Oh, that's fine. Fun, can you guess how many nucleotides to make DNA or RNA?
I think it's 4.
Oh, yeah. Yeah, actually A, T, C, G for DNA, A, U, C, G for RNA. So this could be a little confusing for some of you guys, but so for creating a macromolecule such as DNA or RNA, you have to think what are the alphabet of this molecule and how many alphabets are there. For DNA, RNA, there's only 4 for each. ATCG, adenine, guanine, cytosine, thymine, or AUCG, adenine, uracil, guanine, and cytosine. So out of these 4, we actually kind of combine this together. You guys learn math, actually even not math, arithmetic, right? And then we create this vast majority of a DNA and RNA, which composes of our, our body's blueprint, okay? But sometimes we can get a little confused that, hey, what about protein? And protein is a polypeptide chain. And this peptide is composed of amino acid, and that's 20 kinds. So you can see there are a huge number of protein can be made. Okay, hopefully that makes sense. And this one for reference means like for undergraduate students, this would be a little bit of detail, but for graduate students like you, you should know about it, okay? So the phosphate ions in biochemistry. So we've seen this, let me go back to this, nucleotide has a pentose molecule, organic base, there's a phosphate. So what's the importance of this phosphate? It says PO4 minus, and that's a phosphate ion. And this is very, very important in terms of the energy aspect, all right? So I think I just want to go back, move on to the next. And so we want to discuss about the importance of DNA and now storing our genetic information instead. So let me, I think, let me go to open the new part of this. Where is, oh yeah, here. Okay, so let's go to the next part, 3.3. Now let's go. Okay, so this part, let's see. Oh, there's one question. Okay, it's good to have the question over here. What makes DNA more stable than RNA? So just one question, but let's see. So Anho, can you explain your question a little more again?
I mean, because like the human, they use the DNA to store the genetics, but the virus, they use the RNA to store genetics. So it means like DNA will stable more than RNA. So that's why the human, they choose the DNA to store genetics. So why the DNA is like, I mean, the structure of the DNA, was it make them stable more than the RNA structure?
Okay, a very good question. I think this is a very important question, given we all suffer from COVID virus, and that virus, not all virus, but COVID virus is a genetic information of RNA, and we all know that a new variant of COVID, which makes it more difficult for us to develop a vaccine and treat people. And this is giving a good survival advantage of the virus to survive, while our humans, it's hard to, respond and to develop vaccines. So we know that the RNA probably is less stable, and while DNA is our blueprint, we don't want to have a lot of mutation in our DNA. So that makes sense. So you can consider various aspects of it. I think this is a very important, excellent question. So let's see who, hey, Jong-Yun. So someone who worked with the DNA and RNA could, explain this. Or you guys write down your question to get even more points. Jong-Yun, can you guess from the structural point of view? You're a chemist, so you can say from a chemistry point of view. Can you turn on your mic?
Because the DNA structure is the alpha helix, so there are hydrobonds between between the base, and this makes the structure more stable. So it is the point of that why the DNA is more stable than RNA.
Okay, I think that's a good point, that this DNA has this hydrogen bonding. In fact, I think you want to say DNA is a double strand, which is complementary each other. So if you take off one, you still have that information, and that structure itself is probably more stable than a single-stranded RNA. So everyone should know RNA is a single-stranded, while DNA is actually anti-parallel double-stranded. So that makes it even more stronger. So I think that's a very good aspect. But I don't think that's the only question. So I encourage you, find out and put your answer over here. So the more you have this like 좋아요, a like, and I think we'll consider maybe even giving more points for good question and good answers. All right, so there's more question over here, so that's fine. And
Professor, I answered that question.
oh, excuse me?
I answered that question.
Oh, you answered the question. Okay, let me see. Sorry, somehow I didn't see that. Now, let's continue to study. Okay. Oh, sorry, yeah, great. Oh yeah, there's an answer by Youngmin. If you look at RNA's ribose and deoxyribose, there's 2' hydroxy group. So there's electrostatic repulsion with 3' phosphate group. And RNA sugar has 3' endo-Pucker structure, okay? And DBA sugar has 2' endo-Pucker structure, which is more stable than 3' endo. Okay, it's just the wording is a little bit not easy to grasp. Youngmin, maybe can you explain one more sentence to explain this?
2' endo-Pucker is ribose has Pucker structure because carbon ideal angle is 109.5. So ribose has 5 carbon and have two core structures. So, and 3' end of core is more 2' end of core is more structure because because 3' and 5' phosphate electrostatic repulsion.
Okay. Yeah, okay, good that you have put some answer over here. I'm sure this is not the only answer, but you pointed out very important in terms of structural aspect. So I think what you actually said, oh, that's actually relevant to the next stage. So let's move on. I think we can actually move on to the next stage. Okay, so I put this one, you guys, we are discussing DNA and RNA. I think it's very important to know some of the history of discovery because you guys are supposed to make this kind of history. So this is one-page paper in Nature, 1953 by Crick and Watson. And it's just fascinating if you read this history and you'll just find out fascinating. I just love the story. And there are a number of books over this story, Double Helix, you might have read already. And by Francis Crick, What Mad Pursuit, and Francis Crick, Discovery of the Genetic Code. Let me grab some of the books for you. Yeah. So yeah, I'll just show you this. Just love these books. What a Mad Pursuit. Here, What Mad Pursuit. And this is about Francis Crick. And this is James Watson wrote a huge bestseller for Double Helix. And even Francis Crick wrote Life Itself, The Origin and Nature. I mean, these are all fascinating books. If you are interested, you can come to my office and you can borrow it for a month, all right? So that's something I found it very interesting. And maybe some of this content could be some answer for Youngmin's question as well. So nucleotide basis makes as I say, Watson and Crick, the discoverer of this structure of DNA. Oh, by the way, why structure of DNA is so important? Because we have a lot of questions why we look similar to our parents. Where comes this heredity? Where stores those information? And the answer somehow comes from this nice, interesting structure of DNA. And DNA's structure is a double helix structure, which is complementary to each other. What that means is when the information passed down to downstream, next generation, this information is being stored. And how and why is the question. And you guys probably already know that like nucleotides, they change, polymerize, and that A, T, G, C, T, T, and that information is genetic information we call. However, if it's just single strand, for some whatever reason of you can change this this one letter easily. So that happens in RNA or mRNA, which as we've seen. But with this, exactly the same complementary ones are side to each other. It's harder to make more of this variation or a mutation. So that's structural reason why DNAs probably are more stable, and that's actually necessary for keep our species genetic information. Okay, so that we call hybridization, which is actually involved in each of these hydrogen bonding. Okay, so a single nucleic acid chain can combine with another. Let's see. 이정윤? Yeah.
Another complementary.
Exactly. And what does complementary mean? A goes to T, G goes to C. And there are interesting structural connections so that they prefer each other. Okay, that chain to form a DNA double helix. Why is it double helix? Because it's a double strand, and once it forms, it's interesting. It looks like a helical structure three-dimensionally. Okay, and during this, the process of these two single strand of DNA is assembling in double-stranded DNA, and two strands have complementary sequence such as adenine, thymine, guanine, cytosine. And that process is called, let's see, Yundami, can you guess?
Hybridization.
Exactly, that's called hybridization. So, okay. Okay, so that we call as a Watson-Crick base pairing. And let's get into a little more detail in molecular structure and DNA base. And actually G and A, guanine and adenine has this purine structure, and C and T has a pyrimidine structure, we called it. And somehow inside there's like 3 hydrogen bonding, and this is 2 hydrogen bonding. So that distinguishes these two, all right? So, and then let's move on to the last topic of this section. Is called central dogma of molecular biology. So we ask ourselves the question of, hey, so we are made out of a blueprint of DNA, but our body is made out of multiple structures, including a protein, which is very, very important. Okay, so how does this our protein, an enzyme or structural proteins are made up of from the blueprint? That's an important question to ask, right? So for that, actually Francis Crick actually put central dogma. This is like a little weird because we don't use the term dogma in science, but it's more like a religious term. But that's something like, you know, big guy named it as a dogma, and dogma is a principle laid down by an authority as incontrovertibly true. So if you find something wrong with this, you get Nobel Prize, another one, right? So let's take a look at central dogma as how from DNA to the protein in the end. And we know that from the DNA blueprint, we actually generate RNA molecule, and that RNA molecule making is called transcription. The why we use transcription, you know how transcribe is just 1-to-1, like it's kind of copying 1-to-1 with a rule. So that's why we call transcription. But the RNA molecule, now in RNA molecules, when you open up a dictionary, it's like a word by word. So a number of RNA, the subunits, you interpret as a word and you convert it. So that's why we call it as a translation. And how does it happen? The information flow is from DNA to RNA, and protein is made out of this RNA after translation. Of course, Youngmin pointed out there's a more complicated process of post-translational modification too. So let's hear a process of a translation transcription produces mRNA, and proteins require coordinated chemical reactions, which is catalyzed by enzyme. So we need an enzyme. Enzyme is, by the way, it's also protein, okay? And that's called DNA to RNA. It's called DNA something, and RNA polymer. Let's see, Anho.
Polymerase.
Exactly. So that's called polymerases. And age is usually when you see or enzyme, it's called age. Protein biochemical compounds made of one or more from
Polypeptides.
exactly polypeptide chain, which perform functions in cells and organisms. And DNA to RNA, we already talked about it, translation. And actually, it's not just RNA original, but you have a messenger RNA, which is eventually made into protein. So that we call translation. So I think you guys know this. And let's, let me see. Let's go to the next one and then check that out. Hi, now, okay, so for that, Oh, okay. Okay, Youngmin has one question. I think, let's see. Oh, by the way, we also welcome your question for each section. You write down your question into the chatting. During the class, during people explaining things, you write down your question onto the chatting. I think that would be also good for the class as well. So Youngmin asked, did Watson and Crick expect major growth in the DNA? Okay, how did the Morris Wilkins extract DNA and identify X-ray diffraction pattern? Okay, that's kind of a good advanced question. Hey, so Youngmin, can you explain what is major growth first?
Major growth is region which is binded transcription factor and protein.
Yeah, sorry, it's a little hard to listen, but okay. So I would say Watson and Crick, when they first discovered, I don't think in every aspect they were correct. But the reason why we value them is they made the first correct structure of double helix. So the details. So we know now that DNA is like kind of this right-handedness or left-handedness, and A form DNA, Z DNA, and those were not known at the time, right? So I don't know the answer for this exactly, but someone got just check and then answer in easy term. I think that would be good. And also Youngmin raised a very important question of how did Morris Wilkins, I think it's a Wilkins, extract DNA and identify X-ray diffraction pattern. So I think you guys can answer this, but what I want to point out is what comes X-ray diffraction. We have a DNA molecule which is several nanometer, and we don't have many tools to find out this. So as we say, let's say we use microscope. Microscope uses the light, and the light has only about several hundred nanometer. So we can't really see DNA molecule. We don't have that resolution. So if we want to see even smaller structure, we have to use shorter wavelength, and that's below the visible light. So it goes down to UV and X-ray, and that X-ray diffraction, meaning that X-ray light, X-ray ray interacting with the crystallized DNA, and then the diffracted form. We use our knowledge of the diffraction, and we can deduce the structure, whether that was a helical structure. So this is a very important point you made, and everyone should appreciate that aspect. Okay, so I think that's good. Let's move on to next. So this is, I think, for high school biology, you guys are supposed to know. So I'm just going through with you, let's say genetics and inheritance. And the reason why we put it here is it's very important for us to first, from the history, you guys, we can see from under the microscope to see the shape of the chromosome, which you can see 1.4 micron. So microscopy can actually see this. So to determine, hey, someone got another extra chromosome. Hey, he or she has a problem genetically. For example, famous ones, Down syndrome, right? And then, but once our technology and knowledge develops, we go down to further down to 700 nanometer and coming down inside the chromosome, 300 nanometer. This is probably the end of our optical microscope to kind of look at it. And then from even this single of this chromatin structure, you'll find a nucleosome, which is now 30 nanometer. the beyond the optical microscope. So you probably have to use something like atomic force microscope, which has higher resolution, or electron microscope. I don't know whether we actually uses it. And then you look at this beads on a string form, which is 11 nanometer. I'm actually curious how we actually discovered this. And you go down even 2-3 nanometer of a real DNA double helix. So I do remember atomic force microscope actually shows somehow this DNA structure. Not sure about double structure. Okay, so that's how the blueprint of our genetic information gets up to this chromosomal level. And we know that from cellular division, and we know that these cells divide, and gene expression makes all kinds of 100 kinds of different cell types in our body. So that's our genetics, right? So the genetics is study of heredity and hereditary variation. And we call gene all the time, and gene is actually a location in the genomic sequence, okay? And so genes are located on this chromosome. And within our cellular nucleus, there's a package of double-stranded DNA with, let me see, this is a Fong, can you guess what this is?
I think one.
Yeah. One, yes, right. Histone, yes. Histone protein to form a material called the chromatin. Okay. So all this one, I think you've got to be seen in terms of this nice diagram, and we already showed that from the chromosome, and it's an excellent structure inside. So you can see this nucleosome and histone protein. The histone is like 8 of this, and then you coming down to actual DNA double helix, and this color code actually stands for ATCG, and this one actually represents a kind of a complementary sequence, and actually inside there's a 2 or 3 hydrogen bondings. So that's like the chromosome, and from the history we can use a microscope to find out something's wrong. When some person has some weird characteristics, something's wrong, and we look at like the individual's chromosome, and then we identify somehow some variation. So that we call as a karyotype, and the karyo means like kind of a nut. Okay, so we see from here now the outcome of this we call a phenotype, but that phenotype must come from a genotype. Okay, so that genotype we call genetic makeup of an individual, and the traits which is visible from the outside observed in an individual we call phenotype. I think it's already showed it here. Let's see, chromosomes can be isolated from a cell and displayed in a karyotype. Okay, as how many pairs of homologous chromosomes. So you guys know there's a homologous, which is a pair of chromosome, which is common, but for male and female, there is a sex chromosome. So if that's XX, it's a female, XY, it's a male. So how many pairs of homologous chromosomes? Doreen, can you guess how many do we have homologous chromosomes?
23.
Oh, good guess, but actually it's a 23 homologous chromosome. Okay, and nuclei.
It's what I said.
Oh yeah, that's good, right? The nuclei of all somatic, which is the body cells, contain. So, Doreen again, how many identical pairs of chromosomes?
22.
Exactly, very good job, and a pair of sex chromosomes, which like everyone knows. So now let's go down to this human karyotype. So we know that the gene is really about the location. So we call a locus of gene number 1 and locus of gene number 2. And when there is a genetic trait passed down to next generation, especially this one is in terms of sex cells, and they will undergo meiosis to leading 4 possible haploid cells. So this is we call a diploid. A di means 2. Okay, so human cells that contain, so we already say like 46 chromosomes in total, or 23 pairs containing 2 copies of each chromosome, one from mom, the other from daddy. Okay, so that paired chromosome we call homologous chromosomes, but the reproductive cells such as sperm and egg cells contain now only half of them. So that's why we call it as a haploid. And that cells we call gametes, right? So these are gametes. A father's gamete, mother's gamete. When fertilized, we have this heterozygous or homozygous at the location for gene 1 and 2. So that's we call this physical location of a gene on a particular chromosome we call as a locus. Right? Hopefully that makes sense. And then let's see, human genetics. So this is a little boring stuff. I think you already watched the movie, but let's just go through as a review of this. So each heritable feature, such as our eye color of dark brown for me, and maybe someone has a blue eyes, and that we call, see, a nut. Can you guess what this is?
The color.
Excuse me?
The color.
Oh yeah, it's called, this is called character. I mean, this is a little weird, but yeah, when we call character, this is a little more genetic specific term for that. But that character, when we say eye color, there are variations such as blue eye, red eye, and that variant of character we called a Dorin.
A trait?
Great, yes, that we call a trait. And a gene has the alternative versions of each in the DNA sequence, because DNA is a double DNA sequence. We are using a double sequence in gene that can vary from one chromosome to another. It's called 신소연. It's okay if you don't know. Say no. I don't know. Excuse me. Exactly. That's called a layer, right? Yes. Thus, homologous chromosome is a pair of chromosomes with the same gene, although may possess either identical allele or 2 different alleles. So that's a homologous chromosome. When the alleles are the same, identical, and that individual for the gene is called homo. 이정윤?
Homozygous.
Exactly. That's called homozygous. If they are not identical, that's called heterozygous. So controlling that gene, responsible for that character. So we call about traits, and that set of a trait, which is actually shown. You have blue eyes or red eye or dark brown eyes that is exhibited. So that we call phenotype. However, sometimes even with the same phenotype, the inside gene might not be the same, because there's a 2 is there, and one's dominant and the other is a recessive. So genetic makeup, which may not be always exhibited, but more fundamental inside, is called, of course, a genotype. Okay, so I think that kind of makes sense. So some of the question over here is, let's say, I think I'll just go through this one. So individuals with 2 different layers. Different layer means it's heterozygous. One of the layer may be 정우주.
Yes, Professor. Dominant.
Oh, exactly. Yeah, dominant. or we mean fully expressive. Even one allele is there, it will actually express. So that's dominant. On the other is Uju again.
Recessive.
Exactly, recessive, meaning that without noticeable effect. So, I mean, the last one is like a famous high school level question, but you know, this can come out into your midterm exam. So make sure you understand this, calculating probability of when we mate a male and female, which has either homozygous, which like capital letter stands for a dominant, while this lowercase letter actually stands for recessive. So in this particular case, what would be the probability of outcome? I hope you guys have done this before, and this is kind of important to know. All right, so that's something I want to now go back to that. Questions are here or not? Let's see, 3.5. So let's go. Oh, there's no question over here. 3.6. Hi, now. Okay, there's no question, but you guys are welcome to write question even like during the class and after the class. But by the time when we count, we'll just count by the time, okay? I can't say when is the count, but you're welcome. If I know it's many, so it's likely that you may not have watched it till the end, so potentially. But when you watch, you write down question and answers so that everyone, including you guys, and even, you know, even all the world can benefit from your discussion. Okay, so I think this is the last movie, which, okay, there's a question over here. It's not a question, but Anho made out of the answer for that. Oh, that's okay. Large A, large A 50%, large A, small a 50%. Okay, it looks like similar ones. So now I think I'm going to go back to your chatting, and then let's see what we have so far to discuss. Let's say, okay, let me see. Professor, I have some problem of blah, blah, blah. Okay, that's not relevant to the class. That's fine. Jung-Woo has a question. In case of genetic disease, fixing. So here's a question. Let me stop sharing so that I can see you more clearly. In case of genetic disease, fixing the DNA structure is easy for fundamental therapy. Why genetic therapy in DNA is not active in these days? It's a very, very important question. Okay, so let me, I think this is good time for discussion for that. So there are genetic diseases. I think we'll cover this in the later on, I think probably next, maybe even Wednesday. Fixing DNA structure may be important if there's a disease due to a DNA change, which we call DNA mutation. For example, even single nucleotide, ATCG, even one change can cause a devastating disease. A good example is a, let's say, cystic fibrosis. I have a experience to participate in autopsy of a person who is 54 years old, I think a male, who died at a relatively early age, but because of cystic fibrosis, he has a problem in his lung and the breathing issue. If there's a problem in the lung, many times you die from pneumonia because of the mucus accumulation. You can't fight well with the infections. It's like COVID, right? So when there's infection, you can't fight well and eventually die of pneumonia. But from that devastating, some of the genetic disease, why the question why we cannot do a lot of genetic therapies active in these days. In fact, I would say it's not inactive. It's just a little more hard to treat. So I think it's good to discuss about this, why genetic therapy is not easy. So that would be a good topic. So let me see. That was a Wuju's question. So let's see, Dorin. So you might think of like why DNA change or DNA fixing treatment may not be active so far, but I wouldn't say nowadays I think we are active. Why is it not easy to treat? Dorin, can you guess?
Because it is so small and we do not have tools to modify DNA, I guess.
Oh, yeah, it's very small. I think what your statement was true in the past, but maybe not true now. Now we've seen CRISPR-Cas9, which got Nobel Prize to be able to fixing a single unit of DNA. So I guess this is I think that's why it got Nobel Prize as well. So it may be quite different now. But Dorin's point is and somehow it's important, very small. But I think you would consider that it's a genetic information. So the gene, one single nucleotide has a change. It causes a problem in terms of, let's say, a specific enzyme is not functioning very well. But that happens throughout our body. How can you fix all the cells throughout our body? That's like a question too, right? So let's see. Maybe someone can answer for this again. Youngmin, can you try? Why genetic therapy is not easy? Oh, I can't hear you.
I don't know. I don't know about this.
Okay, that's fine. Okay, maybe I'll ask Anho, who you know about a little more medicine. So from the medical point of view, can you maybe explain why genetic therapy is not easy?
Because like some genetic disease is from the many problem from many genes. It's not from single gene. So for example, if it's made from the single genes, we can easy to for therapies. But if because from the many genes, so it will have many character expression. So I think it's not easy to find out and cannot fit many genes at the same time. And the second list is, I think some genes, they have the problem after they like expression, like environment is will affect the expression of the gene. It's not from the stem cells. The gene have problem is not from a stem cell, but it's from the environment. So I think it's also another thing for therapy.
Okay, Anho made a very interesting 2 points. Okay, the first point is some diseases are polygenetic. So it's not only a single gene change, which potentially will be easier to fix, but it's many diseases are not like that single gene. That is very, very true. I think it's a very good point. Yeah, even single gene defect, it's still very difficult to treat, but many diseases, I think what Anho pointed out is let's say cancer or diabetes mellitus or hypertension. Many we know like kind of complicated diseases involve multiple genes, and it's virtually not easy to fix this many genes simultaneously. That's a good point. And the second one is, can you explain once more about the second explanation?
The second point, I think like some, like when we were born, the genes in normal is not the mutation from the stem cell, but when we affected by the environment after we born, so the gene expression is changed due to the change of the environment. Yeah.
Okay, yeah, so, okay, that's another good point. So I think that's a little bit side of the question because question was about genetic disease, but Anho's point is very important that not all the diseases are from DNA change level, but even environmental like conflict, environmental damage to the genetic information afterwards could cause a disease. Am I right? Okay, so I think somehow the connection is not that great, but one thing I want to point out is how we try to treat cystic fibrosis. As I say, single gene defect will affect all our body parts of all the cells, but some parts, for example, like lungs, are affected. The problem is we have the tool to fix the gene. Let's say we can make a drug for that. How can we deliver the gene into each cell and to fix the genetic problem? And that's another very interesting, it's more like engineering problem. I hope we can understand that aspect as well. So this is now 10:14. I think this is good time to finish. And so we will continue our discussion from the remaining videos. So I strongly encourage you write down your questions and answers in the YouTube, which I think which makes our class hours spending more of discussion rather than just read out our like pre-made slide, which you already have seen. All right, I think this is good, and I'm gonna try finish our class before 10:15, so that's a good time. Thank you, I enjoyed very much, and I hope you guys have enjoyed it well. So also, make sure you write down your questions and discussion even in this YouTube. Hopefully, we have a more lively discussion. Okay, so I'm gonna finish now. Thank you for your attention. Bye.
Thank you, Professor.
Yep.
Thank you, Professor.